The overall goal of this procedure is to utilize high-throughput RNAi screening to identify host factors involved in virus infection. This is accomplished by first depleting genes of interest by RNAi in 384 well plates. Next, the cells are challenged with the virus of interest, stained for viral proteins, and measured for viral infection by immunofluorescence microscopy. Finally, automated image analysis is performed to identify candidate genes for validation and further study. Ultimately, results can be obtained that show changes in infection through quantification of the percentage of infected cells using immunofluorescence microscopy. This allows for the discovery of novel cellular factors involved in viral infection.
Though this method can provide insight into virus-host interactions, it can also be applied to the study of any system that would benefit from using a visual readout of the cellular process of interest.
Individuals new to this method should expect a steep learning curve because achieving reproducible infections while minimizing variability requires considerable optimization.
To ensure reproducibility of results, it is essential to use a single lot number for each reagent used in an entire screen. Grow S2-DRSC Drosophila cells in complete Schneider's medium at 25 degrees Celsius. Cells should be in log phase for experiments. In a laminar flow hood, dislodge the semi-adherent cells from the flask by rigorous pipetting. Count on a hemocytometer. Transfer 125% of the cells needed for the experiment to a 50 milliliter conical tube. Pellet at 300 G for 5 minutes.